Welcome back to the deep dive. Today, we are taking on a source that is, well, truly foundational. It's the essential architecture of human life.
It really is.
We've been diving into excerpts from C.C. Chatterjee's Human Physiology, 12th edition. This text is just a bedrock for understanding clinical medicine.
Absolutely.
Our mission here isn't just to, recite facts from a textbook. We want to synthesize them.
Right.
We're aiming to give you, whether you're a medical student, a young professional, or just deeply curious, the core integrated physiological mechanisms you really need to understand the body as a single interconnected machine.
And we're going from molecular signals all the way up to complex thought.
Exactly.
That holistic view is, I think, the most important part. We're going to build this knowledge from the ground up. We'll start with the secret molecular language of communication.
The world of hormones.
Yes, hormones and second messengers, which really sets the stage for everything else. Then we'll look at the master controllers.
The pituitary and adrenals.
Specifically, their feedback loops and what happens when they go wrong. And then finally, we'll get into how we decode the outside world, our special senses, and then how the cerebral cortex handles things like learning, memory, and language.
It's a huge journey.
It is a comprehensive look at the machine in motion.
Okay, let's unpack this molecular language. Hormones I think they're often overlooked because they're so tiny, but they're incredibly powerful.
Oh, absolutely. They're produced in minute quantities in one place, but act somewhere else entirely.
And they're controlling everything, growth, metabolism, and maintaining that stable internal environment we call the milieu interne.
And what's really fascinating, and I think this is the key point, is that for many hormones, they don't even enter the cell to do their work.
Right.
Think of the hormone as the first messenger. It's like the CEO. It arrives at the cell surface, the factory door, and just sends a message.
Like an encrypted email.
That's a great way to put it. And the cell's internal system, the second messenger, is the one that receives the signal, interprets it, and then executes the changes inside.
So it's translating the message.
It's transmitting the signal within the cell.
Mm-hmm.
The classic examples we have to know are CAMP, CGMP, IP3, and DIGI.
Let's spend a moment on cyclic AMP, or sham-MP, because it just seems to be the most common and versatile one. Can you walk us through its life cycle in the cell?
Sure. So it all starts when a specific hormone binds to its unique receptor on the outside of the cell.
Okay.
This binding immediately activates an enzyme called adrenal cyclase, which is just sort of waiting there on the inner membrane.
Lying in wait.
Exactly. Adrenal cyclase then grabs ATP, our cellular energy currency, and converts it into cyclic AMP. And that's the moment. That's when the signal translates from external to internal action.
And once that action signal goes off inside, what happens then?
Well, CNMP immediately activates a key enzyme called protein kinase. Think of this kinase as a construction foreman.
Okay.
It catalyzes the phosphorylation of other enzymes inside the cell, which basically just means turning them on or off. This then dictates the cell's response.
So it could be speeding up metabolism or...
Right. Or initiating lipolysis, or even controlling gene transcription right in the nucleus. It's a massive cascade of events, all started by one single molecule.
I love this next connection because it means my coffee habit is directly impacting my molecular signaling. There's a brilliant practical application here.
There is.
CAMP levels don't just hang around forever. They're rapidly destroyed by an enzyme, phosphodiesterase.
Right, the cleanup crew.
But this enzyme can be inhibited by methylxanthines. So when you drink coffee, the caffeine, it's a methylxanthine, it actually stops the cleanup crew.
It does.
Which means you get much higher sustained COSI-Phe levels whenever adrenal cyclase is activated. You've basically just hacked your body's response.
Exactly. It just shows how delicate that balance really is.
Yeah.
Now move into a related player, cyclic guanosine monophosphate, or CGMP.
Okay, so similar but different.
Very similar role. It's derived from GTP, and it also activates intracellular protein kinases.
But the factors that rely on CGMP are really clinically important, aren't they? Who are the main messengers that use it?
The primary ones are nitric oxide and atrial natriuretic factor, or ANF. Nitric oxide, which is a powerful local signal, activates soluble guanylate cyclases inside the cell.
And ANF.
ANF, which regulates blood volume, activates membrane-bound guanylate cyclases right on the cell surface.
And physiologically, if I'm thinking about the most significant role CGMP plays, it has to be in vascular health.
Absolutely. Its main effect is to phosphorylate smooth muscle proteins. When this happens, often through nitric oxide release, it causes smooth muscle relaxation.
which leads to vasodilatation.
It literally opens up the blood vessels, a vital mechanism for regulating blood pressure. And beyond that, cGMP is also essential in some sensory pathways, specifically phototransduction in the eye and signal transmission in our olfactory receptors,
So seeing and smelling.
helping us see and smell.
Okay, before we leave the world of hormones, let's look at the glands that make them, starting with the master regulator, the pituitary.
The hypophysis.
Right. Anatomically, the sources detail two major divisions: the glandular adenohypophysis and the neurohypophysis. Now, the detailed parts, pars histalis, pars tuberalis, are important for anatomists, but let's focus on the clinical side, when things go wrong.
And the clinical impact of the pituitary is huge. When we look at hyperpituitarism in adults, so excessive growth hormone secretion after the growth plates have fused.
After epiphyseal union.
Yes. We get the dramatic presentation of acromegaly.
And the physical characteristics are so striking. It gives rise to this really recognizable phenotype. It includes that increased thickness of the lower jaw, prognathism it's called,
Right.
and this massive enlargement of the hands and feet. It's often described as a gorilla-like appearance. But this isn't just cosmetic, is it?
Far from it. This condition is dangerous because the enlargement is systemic. The internal organs involved in metabolism are also hugely affected. The heart, lungs, liver, kidneys, pancreas, spleen, they all get bigger.
And the metabolic effects.
They're severe. We see hyperglycemia, reduced sugar tolerance, and a massive increase in basal metabolic rate, which often shows up as excessive sweating. The takeaway is that this doesn't just make someone tall or disproportionate, it messes with nearly every metabolic process.
Shifting to another powerful endocrine organ, the adrenal glands. Sitting right on top of the kidneys, we're focused on the cortex, which is organized into three layers.
The zona glomerulosa, zona fasciculata, and zona reticularis.
And a critical disorder that comes from that outer layer, the zona glomerulosa, is hyperaldosteronism.
Often called Conn's disease. And this is typically caused by a tumor that's just pumping out excessive aldosterone.
And aldosterone's job is to retain sodium and get rid of potassium.
Exactly.
So if you have too much aldosterone, what are the clinical signs that just jump right out?
The clinical features are a classic set. First, hypertension, because of all this sodium and water retention. Then, muscular weakness, which is caused by the potassium depletion. And then, critically, the metabolic state. The body loses potassium and hydrogen ions, which leads to metabolic alkalosis.
And if that gets bad enough...
If that alkalosis becomes severe enough, it actually decreases the ionized calcium concentration in blood, which it manifests as involuntary muscle contractions or tetany.
Wow. So understanding that whole pathway tumor to aldosterone to potassium loss to pH shift to tetany is essential for diagnosis.
It is. It's a perfect physiological cascade.
And just a quick note on the adrenal medulla, the inner core. It's made of chromaffin cells that secrete epinephrine and norepinephrine.
Yes, and what's unique is its nerve supply. It consists entirely of preganglionic medullated fibers. It's basically a specialized modified ganglion of the sympathetic nervous system.
Now we get to transition from hormones to the nervous system, looking at the very rules the body uses to interpret reality.
Right, the fundamental principles of sensation.
We start with two foundational laws. First, Muller's law of specific nerve energies. This is a highly organized system. It means each receptor organ and its nerve can transmit only one type of sensation.
Yes. In practice, that means a pressure receptor's impulse causes a sensation of pressure, period. It doesn't matter if you stimulate that nerve with electricity, heat, or actual pressure.
The brain only registers pressure.
The brain will only register pressure. If you stimulate the optic nerve, you see light. The modality is fixed by the wiring, not by the stimulus.
And linked closely to that is the law of projection. This is the one that really grounds all this theory in clinical reality.
It really does. The law of projection states that when any part of a sensory pathway is stimulated, the receptor, the nerve, or the cortex in the brain, the conscious sensation is always referred back to the location of the original receptor.
This is why phantom limb pain exists.
Exactly. A stimulation in the brain's hand area makes the person feel pain in their missing hand because the brain projects that signal right back where it thinks the hand should be.
Let's apply this to the sense of hearing. Our range is just enormous, from 20 to 20,000 cycles per second. And we have to understand the boundaries.
We do. The threshold of audibility is the lowest intensity that just barely gives a sound sensation. And if the intensity gets so high it causes actual pain, that's the threshold of feeling.
Now, for clinical testing, we use these principles of conduction. The sources highlight Weber's test. It's a simple tuning fork test.
A very elegant test.
Yeah.
You place the vibrating fork on the vertex, the midline of the skull, and you ask the patient where they hear the sound.
Now, wait, explain this. Why does the sound lateralize or seem louder in the diseased ear in conductive deafness? That seems totally counterintuitive.
It sounds backwards, but the physics explains it perfectly. In conductive deafness, like from middle ear fluid, the sounds from the external environment are blocked. So when you put the tuning fork on the skull, the sound is conducted directly through the bone to the cochlea, bypassing that defective middle ear. Since the external noise that usually masks the bone-conducted sound is absent in that diseased ear, the bone conduction is perceived as being much clearer and louder there.
That's a fantastic clarification. Okay, flipping to the sense of vision. When we shift our focus from far away to a near object, we execute the near response. Accommodation.
Accommodation is a trio of simultaneous reactions. You have to have pupillary constriction, which is meiosis.
Right.
Then you need convergence of the optical axis, so your eyes turn inward. And finally, the accommodation of the lens itself.
How does that happen?
The ciliary muscle contracts, which paradoxically causes the zonual fibers holding the lens to relax.
And the lens just bulges.
The natural elasticity of the lens allows it to bulge, which increases its refractive power for near vision.
And just finally on this, the incredible speed of the light reflex pathway, which controls that pupillary constriction.
It's a complex arc, but we can break it down. First, input. Visual impulses start at the rods and cones, travel through the optic nerve, relay. The key is that fibers diverge from the main visual pathway and end in the protectal area of the midbrain.
Okay.
Then the output command. From there, a relay crosses over to the Edinger-Westphal nucleus, which is the parasympathetic part of the third cranial nerve. And the final action is those fibers relay in the ciliary ganglion, and the postganglionic fibers make the sphincter pupillae muscle contract,
Causing meiosis. The speed of that sequence is just astounding. Now, let's move into the complexities of the central nervous system. Cognition.
higher functions.
Learning is defined simply as the process by which an individual changes behavior through experience. And while we think of this as a brain function, it can happen at subcortical and even spinal levels.
A classic model for this is the conditioned reflex. The sources really emphasize that a conditioned reflex always develops on the foundation of an unconditioned one.
And timing is everything.
Timing is key. The unconditioned stimulus has to either accompany or just precede the conditioned stimulus for that association to stick.
And what about when a learned behavior stops? The concept of extinction.
Extinction happens if that conditioned stimulus is repeated over and over without the reinforcing unconditioned stimulus.
So the dog hears the bell, but no food comes.
Exactly. The reflex becomes extinct. Physiologically, we think this is a state of cortical inhibition just overriding that learned response, like repeated disappointment. The brain eventually stops expecting the reward.
We also have to look at language, which is typically lateralized to the left hemisphere. Damage to specific regions causes very distinct failures, aphasias.
Let's contrast the two major types. First, expressive aphasia or Broca's aphasia. This is damage to the anterior left hemisphere. The key feature here is effortful speech,
It's halted, fragmented.
delivered with great difficulty. But, and this is crucial, the patient's comprehension is still quite good. They know what they want to say, they just can't get it out smoothly.
Now contrast that with receptive aphasia, or Wernicke's aphasia. This is damage to the posterior left temporal cortex, near the auditory cortex.
Right. And here, the speech is fluent, often very fast, but it's incoherent. It's like a word salad. They struggle to understand the meaning of their own words or anyone else's.
So they can't self-correct.
They can't. It's a breakdown in processing the meaning of language, not in producing the sounds.
Finally, let's wrap up our nervous system discussion with the limbic system. Why is this ancient set of brain structures so vital for who we are?
The limbic system is really the central hub for learning, emotion, and motivation. It's deeply involved in memory formation, particularly episodic memory, mostly through the hippocampus.
And it's our emotional center.
It's the seat of our emotional reactions, processing fear, aggression, reward. And through its deep connections to the hypothalamus, it's constantly regulating our essential autonomic processes, keeping us stable while we react to the world. It's where thought, feeling, and bodily control all merge.
We have truly covered the spectrum today. I mean, from the tiny molecular communication of CMP and CGMP,
All the way through dramatic endocrine dysfunctions like acromegaly and Conn's disease.
and right up to how the brain executes the fastest reflexes and manages these incredibly complex cognitive functions.
That synthesis is the true deep dive. Understanding physiology means recognizing that the molecular mechanism causing your blood vessels to dilate is part of the same integrated system that processes what you hear and stores your memories. It's all connected.
It is. And to reinforce this incredible breadth of clinically relevant knowledge, we have a short challenge for you as you reflect on all this.
Okay, so consider male reproductive health,
Hmm.
which is deeply regulated by hormones and environment. The clinical condition of an undescended testis is called cryptorchidism, based on the fundamental physiological requirements for healthy gamete development, which we talked about in the context of maintaining that milieu internal. Explain the specific cause for sterility that we see in a patient with cryptorchidism.
Hmm.
Think about where the testis should be and the environmental factor needed for sperm production.
A great question. And as we close this deep dive, consider this final provocative thought linking the endocrine world to modern pathology.
Mhm.
We noted that hormones like growth hormone can stimulate the growth of certain cancers, like breast carcinoma. This recognition that hormones play a role in disease was a profound shift in medicine.
A huge shift.
It led to the Nobel Prize-winning work of Charles Brenton Huggins, who showed that by manipulating hormone levels, like removing testosterone sources, you could control and slow the spread of certain cancers, like androgen-dependent prostate cancer.
Just an incredible insight.
So think about the implications. A substance that's designed for growth, a hormone, can become the key to treating or controlling a pathology. This intersection of endocrinology and oncology remains one of the most dynamic areas in medicine today.
The more deeply we understand the normal control mechanisms, the better equipped we are to fight disease when those mechanisms go wrong.
Thank you for joining us for this deep dive into the clinical foundations of human physiology. We'll be here next time for another round.